Regenerative approaches to treat cardiac issues.
Treat patients with acute liver failure.
Aging diseases, immune senescence, cardiovascular diseases.
Muscle weakness, sarcopenia.
Digestive problems, the colorectal cancer.
Extending that tissue maintenance.
Providing us model systems.
In vitro platform.
Embryo-like structures.
Stem cells, by their nature, are able to give you different cell types. The hope in the future of being able to, like animals, salamanders, you cut off a limb, they can regrow the limb. Wouldn't it be lovely if we could do that? One of the obvious examples is heart attacks. More and more people are happily surviving heart attacks these days. But you're left then after that with impaired cardiac function. So your heart isn't able to pump blood as it effectively can, so you can't live a normal life. An area that I really think in the future would benefit from this type of stem cell research is to be able, regenerative approaches to treat these type of cardiac issues.
So we work now on trying to understand the mechanism that allow liver development, liver regeneration. We really hope that in the next 5 to 10 years, we'll be able to deliver new cell-based therapy that allow to treat patients with acute liver failure, and in the future also patients with chronic liver disease.
One area that I would expect to see big breakthroughs is in the field of aging. It's not just the formation of our body that's important, which is critically important, but it's the maintenance. There are pathways that can control tissue maintenance. Just to give a simple example, there are mammals that live over 200, and there are others like field mice that live less than 2. Those different strategies were, basically beneficial for reproductive success. So if we could understand these pathways that are important for tissue maintenance and for stem cell maintenance, we could try to basically modulate those pathways in much the same way that evolution has modulated them, and with the goal of extending that tissue maintenance.
Failure of a muscle stem cell to divide and differentiate when it should means that the muscle doesn't get repaired. So a typical disease condition of old people is muscle weakness, sarcopenia. And it's a cause of frailty and falls and inability to do the activities of daily living. So if we could get the stem cells to behave themselves and regenerate the damaged muscle, it would make a huge difference to people's lives. Broadly, stem cells really misbehave during aging. They can do what I've just described for muscle, but they can also differentiate and divide when they shouldn't and give rise to the wrong kind of progenitors. So I think there's a lot of hope now that we understand so much about stem cells because of the work on their role during development, that we might be able to fix some of the things that they do wrong as we get older.
One of the systems that we have been more successful with is the hematopoietic system, and we have been working on rejuvenating hematopoietic stem cells to improve several of the features that contribute to aging of this system and also to aging of the whole organism, like immune senescence. And we hope also to possibly expand these for the application into other diseases like cardiovascular diseases.
We study intestinal stem cells, how they age, how they epigenetically age, and how this can impact the functionality of the intestine because of loss of regeneration, and then we have more inflammation, more infections, digestive problems, diarrhea. And another problem in the intestinal system is the colorectal cancer that still can impact around 4% of the human population.
Stem cells are really, really powerful in providing us model systems to understand normal physiological processes.
One of the key developments that we are looking at is this development of in vitro platform. So the goal now is really to understand at the individual level what is the best treatment, what is the best level of drug, and it's really this personalized aspect I think that stem cell can give us, because we can now derive stem cell from almost anybody now.
Most of the pregnancies fail, and this failure happens most of the time within the first 2 weeks since fertilization. So we try to understand what it is that human embryo has to accomplish within the first 2 weeks. More than 10 years ago, we have created embryo-like structures from different types of stem cells. And those models are three-dimensional life models that are not identical to embryos, but help us to understand how embryos take those decisions, how different factors influence development of those embryo-like structures, and we can follow development of those structures in the laboratory, which help us to understand the questions about the mechanisms of development.